My name is Dennis Vaccaro. I'm a member of the council. Can everybody hear okay? And I'd like to just read a brief introduction. I'm happy to be here with you today to introduce Associate Professor Young-Pearse, a 2004 PhD from Harvard Medical School and Associate Professor of Neurology at Harvard Medical School, as well as the Selkoe Distinguished Chair in Neurology and Vice Chair of the Basic Research in Neurology at Mass General Hospital. She received her undergraduate degree from Skidmore College in 1997 and her PhD in genetics from the Biomedical and Biological Sciences program, also known as BBS. At Harvard Medical School. She established her independent lab in 2010, which focuses on the identification of mechanistic causes of neurodegenerative and developmental disorders of the nervous system, with the goal of identifying novel targets for therapeutic interventions for these diseases. Please join me in welcoming Professor Young-Pearse to begin her presentation, Unraveling the Molecular Roads Leading to Alzheimer's Disease to Enhance the Design of Targeted Therapies. Welcome.
Thank you. Thank you, Dennis, and thank you all for being here today. I want to begin just by saying that, what a difference Harvard has made to me in GSAS. I'm a first-generation college student, and Dennis mentioned I went to Skidmore College. That's in my hometown, where my mom worked as a child care person, and because of that, I was able to have free tuition. And I really hadn't left New York, the state of New York, very many times at all before I came to Harvard as a graduate student in 1997, and it was really quite intimidating. But because of the support systems at GSAS, the wonderful faculty, the wonderful students, it's really been transformative for me to be able to follow my dreams to study neurological diseases. And so I just want to thank the GSAS program and let you know that I'm incredibly honored to be here today to tell you about our work. And so I want to begin by saying, in my research lab at Mass General Brigham, we have Harvard graduate students from GSAS, we have Harvard undergraduate thesis students, postdoctoral fellows, senior scientists, and research technicians who are all working tirelessly to do exactly what we're saying up here, unravel the molecular roads leading to Alzheimer's disease so that we can design better therapies. And to do that, we're really deeply following humans, large cohorts of individuals with aging that are deeply phenotyped clinically over time, over their age, and also molecularly. And what we're doing that I'm going to tell you about in the large part of this presentation is that we're generating these genetic avatars, these living cellular avatars of each of the individuals that we see in the clinic and that we're following, so that we can then make those cells. You make living brain cells in a dish, some of which these images are shown here, are all these living brain cells that we've generated in the lab to try to understand neurological diseases. And today I'm going to focus on Alzheimer's disease, but the principles that I'm going to talk about are relevant to any neurological disease and any neuropsychiatric disease. And we have projects studying neuropsychiatric and neurodevelopmental diseases as well. So I want to begin by first asking this group of people, how many of you are in or have been in the field of biomedical science, or doctors, nurses, PAs, biomedical research? Some, because I want to know who not to let ask questions. No, that's great. No, I think that's very helpful. And also I wanted to ask, how many of you have known somebody or know somebody who has dementia? Almost everybody. And dementia is it's not surprising that there's so many hands. Dementia, by the age of 85, almost half of our population will develop dementia. And dementia is incredibly devastating. It takes who we are as people away from us. It is incredibly painful for family members to watch and of course it's also a huge economic burden on our society, which I'm not going to talk about today. So we really want to, my talk today is going to very briefly at the beginning, in the first 15 minutes or so, we're going to talk about the past. We're going to talk about the historical discovery, how we got to where we are today in terms of current treatments. Oh, I just realized I don't have to talk into that microphone. Is that true? Can you still hear me? Yeah. Okay, great. Great. The historical discovery of how we came to what we have now for treatments for Alzheimer's disease. And which, as you may know, those of you who know people with dementia, they aren't that great. Like, we're not stopping Alzheimer's disease in its tracks like we want to do. We're not preventing it yet. But then in the majority of the talk, I really want to focus on the exciting future and where we are now. It's really a new era for discovery in neurological disease. And I want to describe to you some real data, but also some concepts of why I believe that in the next 10 years, we're really going to see an explosion in new therapeutic interventions for neurological diseases. And so I'm going to begin, I'm not going to go through every year of history here, but I want to start with the first description of Alzheimer's disease formally by Alois Alzheimer in 1906. Of course, Alzheimer's disease existed well before the 1900s, but he was the first to molecularly define the disease in some way. And so in his first description, he described his patient, Auguste D., and described her as she's a 51-year-old woman who had presenile dementia, which means it was before the age of 65. And this is an excerpt from an interview, from a clinical interview that he performed with Auguste. And I think it really highlights the devastation of Alzheimer's disease. And so he begins by asking, what is your name, Auguste? Family name, Auguste. What is your husband's name? She hesitates and finally answers, I believe, Auguste. Your husband? Oh, so. How old are you? 51. Where do you live? Oh, you've been to my place. Are you married? Oh, I'm so confused. Where are you right now? Here and everywhere. Here and now. You must not think badly of me. Where are you at the moment? We will live there. Where is your bed? Where should it be? And then around midday, while the person is eating pork and cauliflower, he asks, what are you eating? Spinach. What are you eating now? First I eat potatoes and then horseradish. Write a 5, she writes a woman. Write an 8, she writes Auguste. And while she is writing, she repeatedly says, I have lost myself, so to say. And this really highlights, I think, the devastation that Alzheimer's disease can cause in taking away who we are as individuals. And what Dr. Alzheimer did back in the early 1900s was he was one of the first to describe what he saw in the brain of this patient after her death. And he described what he saw, and I'll show you some images in a few slides. He's described plaques and tangles in the brain, which are not normally present in an aging brain or at any age. And we now recognize that this person had an early onset form of Alzheimer's disease at the age of 51. And so, as you probably know, AD begins very insidiously, which means very slowly, usually with decreased memory and disorientation, and then progresses for a number of years, and different numbers of years, depending on the individual. With the progression of outward clinical presentation, there is also the progression of pathology in the brain to different regions. So it starts off with mild problems, coming up with the right word, challenges performing tasks, forgetting recent events. It then progresses as the pathology progresses from one part of the brain to the other, to confusion about location and time, forgetfulness of events or personal history, sometimes changes in sleep and sometimes changes in personality, until finally the pathology is spread throughout the brain, which is quite severe. And you see problems, very serious problems that can affect the ability to go on living. And so what Dr. Alzheimer saw in the brain, these are some drawings from him, of his patient brains, were these abnormal accumulations of plaques, these extracellular accumulations of proteins, and then intracellular, what he called tangles. Let's see, where's one? Intracellular accumulation of tangles inside neurons that he called tangles, that are composed of tau. I'll tell you about in a second. There also were accumulation of lipid droplets throughout the brain, and then also these glial cells that were hypertrophied, which means they changed their shape and it was a sign of inflammation in the brain. And altogether, this is what we see in the brain today. So we have more advanced methods for imaging these different pathological features. But what's really specific to dementia and to Alzheimer's dementia are these plaques and tangles. So those lipid deposits and the inflammation of the glia are more generalized. And we can see that in a number of different neurological diseases. And so these plaques are stained here in brown, are composed primarily of a protein we now know is A-beta, or amyloid beta, and these tangles are composed primarily of a protein called tau. Now, I'll say that dementia generally is an umbrella term, and Alzheimer's disease is about 70% of cases of dementia are Alzheimer's disease. And Alzheimer's disease is defined clinically and pathologically by the accumulation of these two hallmarks, A-beta and tau. Now, that other 30% of dementia can result from vascular problems, vascular dementia, but most of those remaining are from a tauopathy, or the accumulation of these tau tangles in the absence of A-beta. So when I'm talking about Alzheimer's disease, and in the coming slides, the insights that we learn about Alzheimer's disease and related dementias, I'm just not continuing to say and related dementias, but a lot of the mechanisms at play and some of the treatments could be overlapping among all types of dementia. And so this is an image from from Cindy Lemire and Dennis Selkoe in our department. And this is an image of a brain, a section of a brain that's riddled with plaques. These brown stains are those A-beta plaques. And what was remarkable that was discovered in the 80s was that there was a lot of people with Down syndrome, people with Down syndrome invariably have brains that look like this. So this is an image from a 12-year-old boy with Down syndrome. And this is unheard of. Without trisomy 21 or even with early onset mutations that cause Alzheimer's disease, it would be very normal to see an accumulation of plaques like this in a young child. And so studying trisomy 21 really gave us a lot of molecular insights into what is happening in Alzheimer's disease. Because on chromosome 21, when there's 3 copies of this, one of the genes on this chromosome is the gene called APP, or amyloid precursor protein. And this is the protein that, when cleaved, makes A-beta. What's helpful, also information and another molecular clue for us, was that not only do people invariably have these plaques and tangles in their brain by age 40, people with trisomy 21, but also about 70% of individuals with Down syndrome develop Alzheimer's by age 60 and 85% by age 70. And the exceptions to this rule, those people with Down syndrome who do not develop Alzheimer's disease are those who have trisomy 21, but APP was not triplicated. So it's a partial trisomy. And that really gave us strong evidence that having just one extra copy of this gene that encodes A-beta was playing a causative role in the disease in these individuals, in Alzheimer's disease. And I'll point out, so here's I should have done this earlier. So there's APP, this is the protein, what the protein looks like. It's a transmembrane domain protein. It's cut by enzymes to generate this little short peptide called A-beta, just 42 amino acids, and that is what's depositing in these plaques. The enzyme, one of the enzymes that's cutting APP to generate A-beta is called presenilin 1. Now, another molecular clue for us for early onset Alzheimer's in the absence of trisomy 21 was that is genetics. So there are hundreds of mutations in hundreds of families around the world who have early onset Alzheimer's disease. And those mutations were found to lie in one of these 2 genes, APP and presenilin, the precursor for A-beta and the enzyme that cuts it. People with these mutations that are listed here, each of those red circles or a colored circle here are different amino acids that are different ones that are mutated in different families around the world. And if you have one of these mutations, you 100% will develop Alzheimer's disease early before the age of 65. So there is these are fully penetrant mutations and dominantly inherited. You only need 1 copy. And so this, again, was really strong evidence that A-beta can cause Alzheimer's disease, at least this early onset form. However, these early onset forms only contribute to about less than 1%. They're very rare. So less than 1% of all Alzheimer's cases have this early onset form. And so, so this was good evidence that A beta was neurotoxic, but following Koch's postulates, one of the things that over a decade of research has examined whether A beta is sufficient to cause synaptic defects or problems to neurons, or whether is A beta neurotoxic. Can we test that in the lab? And so what we and others have done is we can actually extract Alzheimer's disease brain material post-mortem, and we can treat neurons with it. And what we can see is these are neurons in a dish. These are primary neurons. This was done a long time ago now, over 15 years ago. And what we can see is that normally neurons look like this. These are two neurons here. They have these nice processes that connect with one another to communicate. Those connections, they communicate to each other via synaptic signaling, via electrical signaling, and that's how it allows us to think and have memories. And when we add this AD extract, this Alzheimer's disease extract, you can see this degeneration of these neurons in a dish. However, if we take that same brain material and we first remove the A beta from it, now we no longer see that effect. And so this again was good evidence that A beta can be both necessary and sufficient to cause neuronal problems. So, however, so A beta seems like a bad guy, but there is imaging, based on imaging studies, and this is from Risa Sperling's lab at Brigham and Women's Hospital. Based on, we can image the brain in living people for A beta and using PET imaging. And what you can see here is that in this individual on the right, the brighter the signal, the more the A beta there is in the brain. And this is what the signal looks like in someone with Alzheimer's dementia. With someone who's not cognitively impaired, at the same age, in their 80s, you would look something like this. There's some A beta present, but nothing like what we see in Alzheimer's dementia. But then there's this group of individuals who have quite a bit of amyloid accumulation in their brain, but they are not cognitively impaired. And so this suggested that, in fact, maybe A beta accumulation is not sufficient and not causative to the disease, at least in a subset of individuals. And what we can see here is this is one example of one person, but if we look across the Harvard Aging Brain Study, where each of these dots represents the data from a different human, the PET signal for A beta is very high in the AD cases, but there's this set of people who are not cognitively impaired. There's quite a large number of people who have this accumulation of A beta, but somehow their bodies are resilient to those effects such that they do not have cognitive decline in Alzheimer's disease. And so this is a piece of information for you that'll be important as we go on. Altogether, though, there's a lot of evidence that support the idea of targeting A beta as a therapeutic strategy for preventing Alzheimer's disease or for halting cognitive decline. And I'm not going to, I will cover this as we go through, but I just want to point out that there's these early onset forms where we clearly have a problem with A beta generation, and that this leads to the accumulation of tau. We believe that this downstream of this, based on many decades of work, has shown that this can lead to activation of microglia of glial cells, altered tau, widespread synapse loss, neuronal death, and dementia. In late onset Alzheimer's, it's less clear, and this is what I'm going to be talking about when I get to talking about our work. Here, we definitely have A beta accumulating in the brain. It can have synaptotoxic effects, but there are this subset of people who seem to be resilient to its accumulation. I want to point out now here that prior to the 2020s, all of our FDA-approved medications had targets that were way down here on this cascade. So past the point where there was a massive neurodegeneration. So many, many neurons were lost. I showed you that picture of the brain, but where on the Alzheimer's side, you saw that it was quite different than the healthy side. And that's when we were intervening. That's where the molecular stage that we're intervening. Basically, so here it is here. These are the approved therapies for Alzheimer's disease. They're acetylcholinesterase inhibitors and NMDA receptor antagonists. And these are basically trying to save the last remaining bits of neurotransmitters that are present in a brain that has already faced massive neuronal loss. And so not surprisingly, these are not super effective at certainly not recovering function, but also not even in really slowing progression. Many other, not FDA-approved treatments specifically for Alzheimer's, but also many people who have dementia also receive antidepressants and anti-anxiety medications. So then I want to now moving forward in time. that was the state of affairs for many, many decades, was that we had these very ineffective therapies. And then that did not target what we thought or what we think might be the cause of the disease. They were basically managing symptoms of loss of neurons. In 2023, and then soon after, in 2024, we now have two anti-A-beta immunotherapies approved for Alzheimer's disease. These are called lecanemab and donanemab. And so what these therapies do, how do these work? These anti-A-beta immunotherapies, you get these via infusion and repeatedly over the course of it's more than one infusion over time. And what that happens is these antibodies get into the brain, but they also can act peripherally to bind to A-beta and they bind to A-beta, and which is surrounding the extracellular space within the brain and having effects on neurons. And then it recruits immune cells to come and help eat up that A-beta and get rid of that A-beta. And then this means that there's less A-beta in the brain, and that, in theory, would be really helpful to the health of the brain. And so this is some of the results from the first study showing the efficacy of lecanemab in their phase 3 trial. What you can see here are 4 people that were in this trial. This is, again, that amyloid PET imaging. And what you can see is on placebo, the amount of A-beta doesn't really change. But as you have increasing doses of this anti-A-beta immunotherapy, you have this clearance of A-beta from the brain. And that's quantified here over 200 different individuals on lecanemab and 200 on placebo. And what you can see is that this trial was incredibly successful at removing A-beta from the brain. This was really quite remarkable. The amount of reduction of A-beta accumulation in the brain that we observed here, really, like this person who, these higher doses, these people would not even be allowed to enter the clinical trial because they had so little A-beta in their brain at the end. So how was their cognition? Did we save them from Alzheimer's disease? And the answer is, there was some effect, but we could do better. This is not the effect that we would want to see. Of course, what we'd want to see is what we're aiming for. This is cognitive worsening on the y-axis and time on the x-axis. And of course, what we want to see is that with placebo, we don't want to see this, but what we see is on placebo, there's a gradual decline in cognitive ability over time. And what we want to see is that we stop it where it was, right? Once we remove the A-beta, that we no longer have further decline of cognitive ability. And what we saw with lecanemab was that there was a statistically significant improvement, but there was still a reduction in cognition. Who said that? What's the scale? What does this mean in terms of like life? Yeah, and basically what I've heard estimated is, what it would mean is that for an extra year, you might be able to drive, you might be able to live independently compared to previously. But
for all of the behavioral differences,
yes. Yeah, so there's a battery of different cognitive tests that are done, including activities of daily living, which is more of a self-report from family. So there are many different measures. There's many different graphs like this I could show. I just chose to show this one. But I do want to highlight that this was super exciting in that this is the first time we're targeting a cause of the disease and seeing a measurable effect. But in reality, it's not where we want to end up, right? I mean, it's super exciting, but, right? Cognitive decline is slowed, but still declines with time. I also want to point out that this is, as I said, the average, and actually for this graph, this is a much bigger trial than the 200 people per condition I mentioned for PIP staining, for PET imaging. There were 700 people here, and this is the average response. If you look at individual data points, or what we see clinically at Mass General Brigham is that a subset of these individuals respond really well, and a subset don't respond at all. And what you're looking at here is an average, and that's the way that FDA trials are designed if they're not planned ahead of time to subset based on various metrics. So that's going to be a salient point for the way that we're studying Alzheimer's disease, and where we're trying to understand person-specific reasons for their Alzheimer's disease. There may be a subset of people that could really benefit from this intervention much more than what we're seeing here. I also want to point out that the intervention has to be early in order to stop the neurotoxic cascade before you have massive neuronal loss in the brain. And so this is a really important graph that every Alzheimer's talk you go to, you'll see some version of this likely. It's from Clifford Jack. This is from back in 2010, but we still believe this to be true, that this is looking at, on the x-axis, clinical disease stage with increasing severity and biomarker magnitude here. And what we see is that first, if we're measuring A beta in the brain over time, or tau, we're looking at loss of synapses or loss of memory, this is the progression, where first we see an accumulation of A beta, and this occurs 10 to 20 years prior to, this is happening 10 to 20 years prior to someone ever coming into the clinic with a problem, with memory loss. After A beta, then we see this rise in tau. After that, this loss of synapses, and finally the loss of memory. And so if we intervene here, and we can clear all the A beta, it's too late. Like, we already have tau taking off, we've already lost synapses. And so that one message is that the earlier we can go for A beta, but also for other interventions, the better we're off, the more likely it is that we're going to be successful in our clinical trials. And I actually don't have any slides about this, but I think this is a really important point to make. I was talking to some people about this over lunch. The biomarkers for predicting who will develop Alzheimer's disease, blood-based biomarkers from the plasma, that field is exploding. And it won't, and we already they're already being used in research quite extensively to help get people into or not let people into clinical trials, because they're quite accurate now. We can predict even to 10 years before the onset of symptoms who is likely to get Alzheimer's disease with about 85% accuracy. And these are only getting better all the time. So in the past, the biomarker field is exploding now because we have interventions that could actually be helpful. So in the past, the biomarker field kind of lay dormant for a while because people said, who cares? Who cares if we could detect it 10 years earlier? If we can't do anything, then it's like, that's not helpful. But now that we believe that as a field, we believe we're really onto something, not just with A beta, but with other therapies, these biomarkers are going to be critical for success of clinical trials and for success in knowing who to give what therapy in the future. So I do want to say, though, that with these interventions, these anti-amyloid therapies, there is the potential for dangerous side effects. And so it was noticed in this lecanemab trial, and it's even worse in the donanemab trial, this a potential adverse event called ARIA, or amyloid-related imaging abnormality. We see this on MRI, where an MRI of the brain should normally look like this, but you see this white hyperintensity that can occur in different parts of the brain in some people. This is, again, it's identified by MRI. 10 to 15% of people getting lecanemab exhibit ARIA, and people who have a certain allele in the gene ApoE called ApoE4 are much more higher risk. The vast majority of cases are asymptomatic, and they resolve on their own over time. But there is a chance of large brain hemorrhage, and there have been a few people who have died because of ARIA. There's also about 3% of people with ARIA that have headaches, dizziness, and confusion. So it's really critical that when administering these anti-A beta immunotherapies, that people are watched very carefully and that they're selected very carefully to try to mitigate the potential for these very serious adverse events. At Mass General Brigham, we've had no medically serious events after 2,700 biweekly lecanemab infusions across 300 patients. And I think this is in part because our neurology team is incredibly careful about monitoring those patients every time they come in, and also in the selection of people who are allowed to receive this medication. You have to be otherwise, there are many things that could exclude you from being allowed to receive lecanemab. So that's the end of the historical part. Having the first disease-modifying therapy is really good news, but we can do better, and that's what I'm going to talk about in the second part. But I want to give you a chance for just if we can have a couple of quick questions. I have time for a couple of quick questions on that part of what I've already talked about. What are biomarkers? Biomarkers mean just some, what are they? What are they, though? What are the biomarkers? So they're different specific forms of A beta, so A beta 42, A beta 40, and very specific forms of tau as well. That said, we also have a number of other biomarkers that we're interested in that reflect inflammation in the brain. There's and also neurodegenerate, like loss of neurons. So NFL is one of them. There's some synaptic markers we can measure in the blood that also tell us a little bit more about what's happening in the brain.
Do you encounter cytokine release syndrome or something of that sort?
Yes.
That might lead to the kind of...
Yes, and I actually will touch on cytokines secretion and how that can really influence whether or not you decline in the face of A beta. Yeah, that's a big part of what we do in the lab as well. Up here.
When you take the economic studies for a year, have they had measurements that show that some of the tau has been eliminated?
Yes, that's a great question, and yes, that is true. So there's also tau PET imaging, and the tau PET imaging was incredibly encouraging when we saw the decrease in the amount of tau pathology, the progression of tau pathology as well, and then even a decrease beyond the baseline, and also with biomarkers for tau in the periphery and in the blood. So that was actually quite exciting for us to see as well. And then here, and then here.
So have the Down syndrome people been used essentially?
Yeah.
The case where you kind of do it advance, they're going to have a problem.
Yeah, so there's a lot. We've made stem cell models of them, and I'll show you a little bit of data from that. But there's some ethical issues for studying Down syndrome patients, and so they are actually not allowed to get lecanemab currently. And there are a lot of efforts and discussions about whether there should be a trial so they're not traditionally allowed to participate in FDA trials, but they are a group that could be quite beneficial for. And so, no, to date, no, but there's a lot of discussion about this, and they are deeply studied, so that we can get clues as to what's happening. One more question, and then.
Maybe you will talk about that. Do you have a blood test now? You can test it, and here's my money.
Yeah, so I'm not going to talk about it. I probably should have included those slides, I realized, over lunch when I was talking about it. But I do think that there are blood tests that are incredibly useful right now, and there are many academics. It started off in academia, identifying many different blood biomarkers that could predict a little bit here and there, and then putting them together and advancing that over time. Now there's lots of industry. A lot of industry partners are very interested in this because this is something that could be clinically used very broadly. And so that while there isn't right now, I predict in the next 5 years there will be. Yeah, that for sure, probably less than that. Okay, so I want to make sure we have time to talk about the future because I think that's super exciting, but I'll have more time for questions and we can always go back to some of those earlier points later on. And so I want to tell you about why I'm super hopeful for the future and for the current state of affairs, because we're really addressing Alzheimer's disease in new and different ways than we used to study the disease and other neurological diseases. So historically, the way that we study Alzheimer's disease is that we would look at postmortem tissue in individuals, or we do genetic studies from small numbers of people, relatively small numbers of people, maybe 50 people that didn't have Alzheimer's versus 50 people who did have Alzheimer's. Similarly, our animal models were comparing an Alzheimer's group to a not Alzheimer's group. And I'll talk about that in a few minutes. We also have a lack of analytic methods in the past for teasing apart molecular events that are happening in different types of brain cells, and I'll touch on that as well. And so as I said in the previous slide, again, traditional studies kind of lump Alzheimer's disease into Alzheimer's and not Alzheimer's. But in reality, clinicians and neuropathologists have long recognized that there's a lot of heterogeneity in the disease. So there's a vast range of age of onset, even within that late onset group, between 65 and 100. There's different amounts of pathological burden. The amount of A beta and tau in the brain in different individuals varies very widely. The rate of cognitive changes over time changes, and really importantly, the genetic factors that are driving risk and resilience are very different in each individual with the disease. And so, for example, in the cohort of individuals that I'm going to tell you about today that we're studying, there's 2 people, both with Alzheimer's disease, both female. One was diagnosed at the age of 86, but then had a very slow rate of cognitive decline. They had certain genetic risk factors for the disease, whereas this other female diagnosed a decade earlier, and then with a very rapid rate of decline with different genetic risk and resilience factors in that person. What I'm going to tell you about is these human cellular models to study disease. By having that from this range of different individuals, we're able to avoid this lumping together of these 2 people into the same category and expecting them to get the same result. We're able to fine-tune our analyses to look across in a person-specific way why these two different individuals develop the disease. Similarly, in all those individuals that are not cognitively impaired, that do not have Alzheimer's, they're also not all exactly the same. So certainly in our cohort, for example, we have a male who died at the age of 96, no cognitive decline for their age, no A beta plaques in his brain at all, and then had certain genetic profile. We also have a female who is not cognitively impaired, died at 103, no cognitive decline for their age, but had plaques and tangles in their brain that were indistinguishable to a neuropathologist to somebody with Alzheimer's disease. And again, this person had some protective variants in their brain, which I'm going to hit on later on in my talk. But again, we don't want to group these two people, lump them together, and then expect to see statistically significant differences between an AD group and a not Alzheimer's group. I'm sorry, I keep saying AD. That's just Alzheimer's disease. So we've cured Alzheimer's disease many, many times in mice, and that is in part because the mouse models, the rodent models that we use for testing therapies. This is how we test new therapeutic ideas preclinically, is primarily with rodents. But these rodent models, what they are they're very useful, but there's some reasons why there's some downfalls to them. One is that we're basically, again, comparing an Alzheimer's group to a not Alzheimer's group. And these are, the way that we generate these models that have been generated in the past is that we're introducing and often overexpressing one human gene with a mutation that causes early onset Alzheimer's. So a mutation in APP or a mutation in presenilin. These are useful, in wild type mice, there's no A beta plaques, there's normal memory. This is just a little view of whether there would be A beta plaques. Whereas in these mice, they do accumulate A beta plaques in the brain, and they have memory impairment. And so these are the models that we're using. We'll administer anti-A beta immunotherapies or other potential therapeutic interventions, and then we can rescue these memory impairments in A beta in the mouse. But these very rarely translate to the human condition when we go into human clinical trials. And we think that this is in part, while these are very useful, we think that this is in part because we're missing all of that heterogeneity and all those different genetic risk factors and resilience factors that can contribute in humans. And so what I'm going to tell you about with the stem cell model is that we can start to get away from this AD versus not AD. The second point here is that in the last decade or so, there's been an explosion of technologies that are allowing us to look at a finer resolution at what's happening in the brain of people with Alzheimer's and people without Alzheimer's. And I will, and one of those, and I'll talk about those in a few minutes, but we also have larger numbers of individuals that we're studying at once. We have much bigger data sets. So we have genetic studies that include over 1 million people now for Alzheimer's disease, which gives us much more power to detect weak effect sizes, but that give us molecular clues to the disease. We have post-mortem studies where we can measure every single protein in the brain, every single lipid, every single RNA. And we can do this in part in a single cell analysis to uncover cell type specific molecular causes of the disease. And so these studies have really raised a number of new therapeutic hypotheses for why people are developing Alzheimer's and how we might intervene beyond A beta. And so I want to just step back before I get into the cellular model and just briefly highlight the cell types that I'm going to talk about in the human brain. This is an image from the Lichtman lab here at Harvard of neurons. And of course, neurons are the communicators of our brain. This is what's degenerating in the disease. And so it's critical to have models of how these neurons communicate with one another. Glial cells, there's astrocytes and microglia. Microglia are considered the immune cells of the brain. And these astrocytes are really important for maintaining neuronal health. They provide the cells with glutamate so that they can signal to one another. They get rid of some of the toxic factors that are building up. And as I mentioned here, these microglia are the immune cells of the brain. They're always surveying their environment and cleaning up the debris that can happen, including A beta in the brain. So they're also very important cell types. And the vasculature of the brain is also critically important for the health of the brain. And there's a lot of signs, there's a lot of evidence that earlier changes in vasculature, in vascular cells in the brain can affect Alzheimer's disease progression and risk. And so when looking at the brain, not only do we see an accumulation of A beta plaques and tau tangles, but we also have this massive we see this massive degeneration of neurons. We see this hypertrophy, this activation of astrocytes, and this is where these cytokines get massively secreted and influence the surrounding microglia and neurons. And we see these inflamed microglia, which also can not only do their eating up of the bad things that are around, but they start eating up the good things. They start eating up the synapses, which is also problematic. And again, with breakdown of the blood-brain barrier, which can happen in the disease, you can have the infiltration of dangerous molecules that can affect the neuronal signaling. So I want to point out here that contributing to all of these changes that we see in all these different types of cells in the brain there's a number of different factors that can contribute to risk to Alzheimer's disease, but twin studies have estimated that about 65% of the risk can be defined by genetic risk factors. And so, and 35% being modifiable or environmental. And that's a huge percentage that's contributing to risk. And this is something that really we've ignored for many decades, experimentally, I should say, but that we are no longer ignoring. If we break that down, there are those familial Alzheimer's disease mutations in APP and presenilin, as I mentioned, is less than 1% of the population. ApoE4, which many of you may have heard of, it's one of the stronger genetic risk factors for Alzheimer's, but still only contributes a relatively small percent to total genetic risk. And I want to point out that there's a huge amount of genetic risk that is still completely unknown, but that exists in our genomes. And what we want to be able to do is really capture those known and unknown genetic risk factors by having a model to study disease. So this is a complicated diagram about the genetics of the genetic landscape of Alzheimer's. But I want to point out just a few things about this diagram. On the y-axis is the risk for Alzheimer's. So up here are these really super penetrant, fully penetrant mutations in APP and presenilin, but they are very rare. So on the x-axis is frequency in the population. ApoE4 is about 25% of the population has at least one E4, so it's relatively frequent. And the risk to Alzheimer's disease is in the middle. So it's, if you have one copy, you have about a 3 to 5% increase in risk. If you have two copies, it's about a 10 to 15% increase in risk. But for the majority of people, late onset Alzheimer's disease is a contribution of many variants in different genes of small effect size. And so this is, again, incredibly common. So everyone in this room has some genetic risk variants for Alzheimer's disease. I'm fairly confident in that. But it's really the combination of these different genetic risk factors together that add up to pushing us over the edge to developing amyloid plaques, tau tangles, and synapse loss. In this review article, the authors also highlighted by color of these different genes, the different biological domains that have been known to be implicated with those genes. And you can see that there are many. There's APP metabolism, tau, cholesterol metabolism, immune response, endocytosis, et cetera. And so there's many different biological processes also at play. When we look at the cell types that are relevant, that these genes are active in, we can see that there are many different cell types that these genes are active in. This is not just a disease of neurons. It's a disease of neurons, astrocytes, microglia, and endothelial cells. And I want to especially point out this microglial category where there's many of the genes that are primarily active only in microglia or in these immune cells in the brain. Suggesting, and what this really suggests to us is that it's causation. So genetics is the human experiment. Like genetics really tell us what can, genetic association with disease really tells us what are those influences that are causal to risk. And so this really points to microglia, as well as all of these cells, to contributing to causation in the disease, to risk in the disease. And I want to point out that these neuroinflammatory processes of astrocytes and microglia, we really believe that these are happening downstream of A-beta. We know through many different types of experiments that accumulation of A-beta can induce this inflammation of astrocytes and microglia. And those genetic risk factors are making them more or less vulnerable to that effect to ultimately push us over to into the region of accumulating tau and loss of synapses. And so this is the way that we conceptualize the disease, that we have genetic variants and environmental factors that converge in varying different ways on different biological domains, and we see these as different molecular roads that can lead to Alzheimer's disease in different subsets of people. they all converge on A-beta accumulation, tau accumulation, loss of synapse, and dementia, but they do so in different ways. And if we can understand these different ways, we are going to be able to better identify therapeutic strategies for preventing the cognitive decline that happens with dementia. And so human stem cell lines capture these, or what we're able to do with these is we're able to capture person-specific genetic risk and resilience for this and other diseases. And again, these are like, what we're capturing here are the genetics of each of these individuals. These are little genetic avatars of living cells that we can manipulate and study disease in. And so what are induced pluripotent stem cells? The scientists in the room probably already know this, but I want to briefly outline what they are. From individuals, from any person in this room, we can get a blood sample or a skin sample. We can grow those blood cells and transform them to become stem cells. And the way we do this is just by adding 4 genes. So 4 different transcription factors, master regulatory factors that go into the nucleus. And produce proteins that affect the genetic architecture of the DNA, and this allows those cells to be reset to a pluripotent state. So there's 2 key features of these stem cells, of these iPS cells. One is that they're self-replicating, so they divide to make a copy of themselves, and they do this infinitely, which is really wonderful because that means we can have an unlimited supply for testing interventions and for sharing with the world. That's them being pluripotent, or being able to self-replicate, and also they're pluripotent. And that means that if we can use they're capable of becoming any type of cell in the body. And so if we use our knowledge of how these different types of cells normally develop, we can then direct these cells to become those cells in a dish. And really importantly for us, of course, our brain cells. This has been a really transformative finding, especially for neurological diseases where we're unable to take living human cells out of a living person's brain and study them in a dish. And this was such a transformative finding that Shinya Yamanaka won the Nobel Prize for this in 2012. And so I want to point out that the discovery in mice first and then in humans of this technology is about however many years ago that was, but it's relatively new in the scope of things. And at first, it was a rocky start to get to the point where we could actually have stem cell lines produced in large numbers from large numbers of people, and also the ability to generate really good models of the brain from these cells. It took about 15 years for us to really refine, we as a field, to refine and to improve upon these methods. And so now, though, we're at this critical point, I think, in the field where we are able to generate many of the different cell types in the brain from iPS cells, neurons, astrocytes, microglia, endothelial cells, and pericytes. And we're able to put these together in various combinations and in different ways to try to model different molecular aspects of how genetics are affecting the biology of these cells. And so in the last 15 years, as I said, there are a number of different ways that we can use these stem cell models. We can make them into these different cell types. We can look at them in very reductionist systems of single cell types or co-cultures in 2 dimensions. This can give us incredibly helpful insights, which I'll show you about in a few minutes. We can make them in 3 dimensions where we can have more spatial organization of neurons, astrocytes, and microglia. And what I want to briefly show you in a few slides is something that I'm really excited about that we've developed over the past 4 years, which is this brain on a chip model. We've done this in collaboration with Don Ingber's lab at the Wyss Institute at Harvard, where we can take our iPS-derived cells and we can put them in an organized, and we can have an organizational structure, this brain chip, which looks like this. This is a picture from Emulate. So we use the Emulate plastic, the PDMS, in channels, and we use our iPS derivatives to populate this chip. And now this is what it looks like. There's 2 channels, and in these channels, there's a vascular channel where we can line this vascular channel with endothelial cells, pericytes, and astrocytes to make the blood-brain barrier, to simulate the blood-brain barrier. And then we can flow, this goes into a machine where we can provide flow of materials, we can put in blood or media or drugs through the vascular side and through the brain side at different speeds, and deliver drugs and interventions through the vascular component to brain cells that are present in the other channel. And we can study how this delivery of these drugs is affecting the biology and the molecular profiles of brain cells from different humans, from different people across the cognitive and neuropathological spectrum. And this is just a picture of what the cells look like in the brain compartment. We can label each each of the cell types in different colors. And if we focus, this is looking down on the chip, and if we focus through this brain compartment, we can then look at the endothelial cells, which form this nice structure. What's remarkable about the blood-brain barrier is that there's these really tight green lines, these tight junctions that keep the cells really tightly packed. And that's a point about the blood-brain barrier, is that it's very difficult to get things across the blood-brain barrier, including drugs. And this model allows us to study those processes. I have this slide here to remind myself to highlight to you that what's really exciting about this is we have these systems now, these advanced cellular models of disease, and they're living human cells. They're living human brain cells. So we cannot just look at them and say they're pretty. We can actually measure their functionality. We can do various omics profiling, which we've done to get a really strong handle, a large amount of data about all the different proteins and RNAs expressed in each type of cell. We can measure the electrophysiological activity of the cells communicating with each other in real time. We can look at cytokine secretion and how different insults can activate those astrocytes to secrete cytokines and how to rescue that, et cetera. So because of this excitement in these new cellular models, this new era of being able to test interventions and understand disease, the FDA, the FDA Modernization Act 2.0 and 3.0 were passed in 2002 and 22 and 24 respectively. And even the FDA is starting in Congress, is starting to recognize that these cellular models may be useful to test not just safety, but primary, even efficacy, and allow us to bypass, in some cases, animal models to try to test new therapeutic interventions. And I think that this is a really exciting time because sometimes something might not work in a mouse, but it might work really well in a human. If that protein is not present in the mouse brain, that target, you're never going to get efficacy in a mouse. And so now with these two acts, we're moving forward, moving closer to being able to do this in a dish. Oops. So I wanted to just show some pictures of what it actually looks like to do this work. So these are some graduate students and postdocs in my lab working away. It's really quite not that high tech. And when you look at it like this, I mean, if you're not looking under the microscope, basically each of these plates has a different person's brain cells in there in various formats. This is where we have those blood-brain barrier chips. But these individuals work really hard to try to understand the biology of what's happening in the different brain cells. And so I'm gonna skip the questions for now because I want to make sure I get to a few more things before my time is up. So we want to capture these genetic variants across many people. And so in collaboration with David Bennett at Rush Medical Center, we've generated IPS lines now from hundreds of individuals. These cohorts these are the Religious Order Study and Memory and Aging Project, as well as the African American Corps. And here people enter without dementia at age 55 and above. They come in every year for a battery of clinical evaluations. They consent and donate their brain at death. And we have these really rich data sets from all of these individuals, including genome sequencing and omics profiling of the brain tissue of those individuals. So we decided on the first pass to make IPS lines and models from people who are deceased so that we can have the brain tissue from the same people to validate hypotheses that we generate from the cellular model. This is, as I said, a collaboration with the New York Stem Cell Foundation. So we raised the money to generate these lines through philanthropic support. We then convinced David to give up his precious PBMCs for generating these IPS lines. Scott Nagel at the New York Stem Cell Foundation generated the lines, and we got the privilege of being able to make these into brain cells to study their biology. This is data from this is global cognition for each of the people for the first 100 lines that we generated. And what you can see is that here's this person who died at the age of 96 with no cognitive decline. We have people who had, the female that I mentioned earlier, who died with Alzheimer's disease, and then people who had no cognitive decline, but who had high plaques and tangles, and I'll show you that in the next slide. So each bar represents data from a different person, and here's the A-beta and tau metrics for each of those humans as well. So this is that highly resilient category of people. You can see how much A beta, and even for some, how much tau they had in their brain, but were not cognitively impaired in any way. And so we're studying these cells. We also have gone through efforts to try to, the first 50 lines we made, we didn't look at ancestry, genetic ancestry, and they were all over 90% European. So in the next 50 lines, we tried to capture more genetic diversity in these cohorts. We've now done so, and now we can make brain cells from hundreds of different individuals and study processes in these brain cells. And we've also already, because these are stem cells, they divide and self-replicate. We've shared these lines with over 100 labs across the world who are studying not only Alzheimer's disease, but other neurological diseases as well. So in the last few minutes, I just want to show you a little bit of data using stem cells to try to understand disease and understanding these different different biological domains, these different molecular roads to disease. And so we conceptualize the disease again by have a combination of different genetic risk factors that intersect on different biological domains to lead to cognitive and neuropathological traits. So I mentioned early onset Alzheimer's. Very early in my lab, that we developed IPS lines from a father and daughter who both had one of these fully penetrant mutations. They lived on the Cape. They had these mutations in APP. The father had Alzheimer's disease already. The daughter was 35, had not yet developed Alzheimer's, but knows that she's going to develop Alzheimer's disease. And from those cells, this was a decade ago now, in the early days of IPS technology, we were able to make neurons, and we could measure the amount of we could see that they were producing quite a bit of A beta, more in the red bars. These are from the family members compared to controls. They also had higher tau levels here in red. And what was really exciting, even a decade ago, was that we could add anti-A beta treatments, and we could block the effect of the A beta on tau in a dish. And we were able to look across different antibodies to try to prioritize which antibodies were most effective at neutralizing the A beta effect. Similarly, we made lines from Down syndrome individuals, and we studied those, and this was the work of Chun-Yi Wu. I should have said this is the work of my very first postdoc, Christina Muratore, who is still with us today and a faculty member at Harvard. Chun-Yi Wu worked with these Down syndrome individuals, and what Chun-Yi did was, as I mentioned, we have 3 copies of APP. When you have 3 copies of chromosome 21, this leads to high A beta levels. And what Chun-Yi was able to do was go in with CRISPR and just cut out one of those copies of APP. And then what he saw was that when he made these cells into neurons, that then he could rescue a number of different defects in these neurons, including A beta levels, of course, but also tau levels and synaptic defects that we were able to measure in these cells. 2 graduate students, Kazem Terzioglu and Vicky Chow, shown here at her thesis defense, have been studying a gene called SHIP1, which is only expressed and present in these microglial cells. And Vicky's work has shown that these microglia, when you have less SHIP1, you have these inflamed microglia and you have activation of the inflammasome. And what Kazem has shown more recently in trying to understand why you're getting activation of the inflammasome is that if we go into each of these little I should go back. I should explain it better. I'm trying to go too fast. Each of these green cells is a microglial cell. And here, for example, if you look here, what we're looking at is SHIP1 in blue, and we're looking at the protein degradation machinery of that microglial cell in red. And as I mentioned, microglia chew up A beta and they get rid of bad stuff that's happening in the cell. And what Kazem is able to see by using this type of microscopy is that SHIP1 is actually located and may be functioning inside these little garbage disposals of the cell, which is really surprising and completely unexpected. And what was important about these studies is that Kazem and Vicky together were able to show that not only can we see these defects in this cytokine storm that reacts to a lowering of SHIP1, but they're able to rescue those effects by testing different therapies and finding an NLRP3 inhibitor, this is an inflammasome inhibitor, that can block those effects. Zach Auger is a current graduate student who will be graduating this coming year, and working with Yichen Che. They were studying this different type of garbage disposal in the cell. And what they saw was that different neurons from different genetic backgrounds they didn't have the same ability, intrinsically encoded ability, to degrade bad proteins in the cell. And what they found was that proteasome enhancers, so enhancers of this garbage disposal, may have a therapeutic benefit to protect neurons from various, from degeneration. I'm trying to go fast here. I'm going to skip this one because I really want to mention Alex's work. So Alex just graduated last month. She's going to be walking in May. And what Alex found through a series of studies of resilience, of resilient individuals, was this genetic risk factor called CLU, or clusterin, was able to protect neurons from degeneration. And this is related to this cognitively resilient group. I'm going to show this slide, which is it's her rendering of actual immunostaining of cells in a dish. But what it's relaying is that CLU is secreted. What she found is CLU is secreted by astrocytes in the brain. And what it does is, through a series of molecular events, it protects synapses from being eaten up by microglia. What's really important is that in humans with CLU genetic risk variants, she saw that those individuals could not upregulate CLU in their brain. So individuals who had CLU protective variants, when they had high A beta, they increased the amount of CLU in their brain, and those people had very minimal cognitive decline. However, people with these CLU risk variants did not have that benefit, and those people had a very steep cognitive decline. We're really excited about a potential CLU gene therapy that might be a viable option for increasing resilience. And I will briefly go back to this. So this is one of my final slides. So going back to this diagram of the different molecular roads, what I hope I've convinced you of today is that by using these IPS avatars of different humans, looking at different brain cells, we can start to disentangle these different molecular roads and identify therapies that might be appropriate for the causes for each of these sets of people. Anti-A-beta immunotherapies may be very useful for people who have a problem with A-beta generation. I skipped over that one. Inflammasome inhibitors may be very useful for people with immune dysfunction, and specifically those with these SHIP1 risk variants. We have individuals who have defects in proteostasis that we can measure the ability to turn over proteins. We can measure that in a dish, and we can target them with proteasome enhancers, which are being developed by various industries. And with regards to the subset of individuals who have CLU risk variants, if we're able to upregulate CLU in those individuals, we may be able to protect them from the toxic effects of A-beta. And so to summarize, there's a lot of challenges with using stem cells to study disease, but I think there's a lot of potential for these to really have a big impact on our ability to find new interventions and to test those in a person-specific way. And as I mentioned, this is not specific to Alzheimer's disease, but can be applied to a number of different disorders that we are very interested, that we're studying in our department and beyond. And so I want to conclude. I have an acknowledgement slide because I hope I didn't skip over too many people. I tried to show you pictures of people as we went through very quickly there. But I also want to thank all of our collaborators. We have it's something that's changed over time also in the last 20 years since I was a graduate student, is how collaborative science has become by biomedical research. There's a large amount of data present, and there are a lot of advanced techniques, and we need to work with the absolute experts in every component of that in order to move the field forward. I also want to thank our funding agencies, and also acknowledge that the work wouldn't be possible without the participation of human individuals with these diseases and their families, who have really made a big difference in our work, as well as generous philanthropic support from different foundations. And I will stop there and take some questions, and I will be around to take more questions after, so we can do that over drinks if you prefer. But thank you. In front and then up here.
Do you want a microphone? I'm always looking for prevention. So is there any supplement or other things you can do to slow it down?
So I think that all these interventions are things that we would want to do as I mentioned earlier, the earlier you can go in, the better. And so primary prevention trials are difficult trials, right? Because they take a long time to find out what's going to happen. But I think there's no supplement. So there are many people that will recommend various supplements. There's no specific supplement that you can take that is going to protect, that we know of currently, that's going to protect you from Alzheimer's disease. But keeping your entire body healthy makes a huge difference in your risk, in your resilience, I should say, for Alzheimer's disease. But maybe that's not what you're suggesting. But I think that with these different interventions, we do have to think about prevention and not just treating the causes of the effects of the disease.
Thank you.
Thanks for the talk. Do you know how CLU is
is upregulated?
Yeah.
Is it like a transcription factor? And
Yeah.
how CLU is downregulated? Is there like epigenetic modification?
That's exactly what we're looking at. Yeah. So how is CLU upregulated and why is it upregulated in some and not others? So those genetic variants that are associated with Alzheimer's disease, we can see that those are associated with changes in CLU levels over time, both in the blood and in the brain of individuals. We have a lot of ATAC-seq data. You seem like a scientist. We have a lot of ATAC-seq data from the cells and from the brain of individuals with 2 copies of the protective and 2 copies of the risk. And that's exactly what we're looking at. Is there, where in the genome is that key factor? And one thought is to use an antisense oligonucleotide therapy to try to control expression by if we know which part of the genome is responsible for that upregulation.
And one more quick question is the brain on a chip model.
Yeah.
Are you able to grow the cells in 3D, like an organoids on the chip as well?
Yep, you are. Yes, exactly. We wouldn't do that in the vascular side just because we want to be able to flow material through there. But yeah, it's a fairly large region that we could incorporate a 3-dimensional structure. Yeah. What else? Yeah.
Thank you for your talk. I remember attending this event probably 20 years ago, and someone gave a lecture on Alzheimer's and was very confident that understanding the tau and A-beta and all of that, we understand the chemistry, so it shouldn't be too long before we get control of this disease. And that was 20 years ago. And it reminded me of a talk that I heard probably 50 years ago on nuclear fusion. And we're going to have nuclear fusion before too long, not later than 10 years from that date.
Yeah.
Anyway, so I want you to just consider the basis for your confidence, which I hope is the case because my sister-in-law has Alzheimer's.
Yeah, absolutely.
The second question, maybe you could just combine them, is there are intimations that the current government is cutting off Alzheimer's leadership, if not research funding, and how might that affect your program?
Yeah, those are 2 different questions. The first question, I can try to combine them. So about 10 years ago, there is progress from there, right? So we do now have two FDA-approved therapies that target A-beta, and another that's been fast-tracked just last week for tau, antisense nucleotides, to try to knock down levels of tau. So that research, it wasn't right around the corner. It took 10 years, but I think that we have made progress. So I'm going to give my colleagues, whoever they are, I think that there's been progress, and I think that having those disease-modifying therapies is really amazing. I know the benefit doesn't look impressive, but we didn't have that at all before. We had never been able to slow progression at all, and so that is the first step. And now the hope, my enthusiasm is that we can build on that, because the progress has been exponential in terms of the research and the accumulation of data and information about this disease. It hasn't been incremental. It's not this slow up, like we can have to wait another 10 years to see cluster and be able to do something. No, it's really quite dramatic for a number of reasons that I outlined today. And so that's where my enthusiasm lies. In terms of funding, of course, it's a very stressful and anxiety-ridden time in our field where we don't know what's going to happen with NIH. I'll say that our work is about 80% funded by NIH. And the remainder is from foundations and philanthropic support and a little bit of industry. But we have to persevere, and we are going to persevere. We are committed. The researchers like myself and many others are committed to understanding these devastating disorders, and we're going to find one way or another to make it happen. We aren't in it. We've never been in it to make a whole bunch of money, and now we will do what we need to do to diversify our portfolios and lobby for keeping funding for this important disease. But I am optimistic that over the course of time, we will keep moving science forward in spite of whatever might happen in terms of NIH specifically. That doesn't really answer your question, but that's how I'm feeling at this moment. I mean, I think one of the concerns also, though, the biggest concern I have right now is that in the next generation of scientists, my graduate students are super concerned about, they want to stay in science, they want to keep studying, doing research in academia. Some of them want to go into industry, and they are, and that's great. But like one of the graduate students came to me and said, I have to support my family, and I don't know if I can handle the stress of the current financial situation, the current funding situation, which is really unfortunate because she's a super brilliant scientist who can make a huge difference. Oh, we should have a happy question before we end.
One more question before we
Yeah. One more question?
Okay, well, I guess apparently we're at a point in history where all the easy diseases have been discovered and fixed. Now we're left with the complicated, really hard stuff. And I'm so proud and happy that we have people who are willing to spend time trying to figure out these very, very difficult multifactorial things now. So, but I believe we're at a point where we're going to be able to rapidly progress. And okay, so AI, that's all I'll say. But maybe yes, maybe no. But but I really do believe that we have a lot of promise. So I want to thank Dr. Young-Pearse, sorry.
There you go, it's okay.
Yeah, no, that's all right.
You gotta shake my hand.
Yeah.
Shake my-
No. Here's a little present from the Graduate School of Arts and Sciences. Thank you so much for doing a wonderful presentation.